Hello people, this is Dr. Rohan Modi. I'm your Medbuddyy. If you guys are new here, please go through my introductory video to this series to know how to go about all these videos of 10 days preparation. So guys, even before going into what is embryology and what are the steps that goes on in the embryology, let us discuss something known as chromosome. Okay, why chromosome? Why should we discuss about chromosome? Even before going into the embryology, let me tell you about it. Okay, so you've all heard about something known as chromosome, DNA, all these terms, right? So this is the central processing unit, which is the CPU of our human cell. Okay, there is nucleus. Inside the nucleus, there is DNA material. This DNA material in a particular shape is known as chromosome. There is DNA, but why do we call the other term of chromosome? DNA is the structure which is always present in the nucleus in a chromatin form. This is known as a chromatin form of a DNA. But when this cell undergoes a division, this chromatin forms a certain particular shape of a double dumbbell. This shape is known as chromosome. One of the arm of this chromosome is known as chromatid, and two of these chromatids join at the centromere to form the chromosome. Okay, so this is all you need to learn: what is DNA, what is chromosome, where the nucleus is present, etc. Okay, so now that you know that what the chromosome exactly means, so let us learn the structure of the chromosome and what are its functions. There are 46 number of chromosomes inside a human cell, which differs from each and every organism. Okay, the dog has a different number, the monkey has a different number, etc. Okay, so human body cell contains 46 chromosomes. But when we see a textbook image or everything, we tend to see only one chromosome that has been given in a cell, right? This is nothing but the representation of one single chromosome that we are having in a cell. In the textbooks and all, they tend to give only one single chromosome. But in reality, there is 46 number of chromosome. Okay, so there are 46 number of chromosome in a cell or inside a nucleus of a cell. So in total there are 46 numbers. So let us discuss about the structure of chromosome. The structure of chromosome has two different arms. One is P arm, the other one is Q arm. This P arm contains pseudoautosome region, which is also present in Q arm, that is pseudoautosome region. The euchromatin region is also present in both. The only difference here is the heterochromatin region, which is present in Q arm, the SRY gene, which is present in on the P arm. Out of all these things, the main and very important thing that you will have to remember is about the SRY gene. Why SRY gene? Because SRY gene is the gene that gives rise to the formation of male gonad. This is the differentiating factor between a male gonad and a female gonad, whether the sex has to be male, whether the sex has to be female. This is the gene that is present in the chromosome that decides whether the formation of the sex is differentiated. Okay. This chromosome, as I've said you before, this is nothing but a CPU. Okay, this CPU contains all the information the cell has to go into. How many times the cell has to divide, or what the characteristic of the particular cell has to be. Both the chromosomes, that is 23 from mother and 23 from father, has come to form 146 chromosome cell, right? So these are the characteristics. These characteristics of mother and father will be inside that chromosome, which pass on to their upbringing. Okay. So, in chromosome, there are particular regions. As I discussed below, in that 46 chromosomes, there are 2 sex chromosomes and 44 autosomes. Autosomes is which gives the body that particular character. Okay, for example, which color of hair that the baby has to have, what type of nails that the body has to have, what height, etc. All these things will be decided in the autosomes. Okay, but the sex chromosomes, the 2 sex chromosomes, that is XX or XY, will give that particular human body's sexual characteristics. Okay, so this 46 chromosome will be 44 plus 2. 44 the autosomes, the 2 are the sex chromosomes. This is all you will have to remember in a chromosome chapter. Okay, that's all, nothing else. You need to, the questions that come up here in the chromosome related topic are where does the SRY gene present? The SRY gene is present in the P arm of the chromosome. What joins the two chromatids of chromosome? Centromere is the main thing that joins the center between two chromatids. That's it. Nothing else. You need not remember about the heterochromatin region or the euchromatin region, the pseudoautosomes, etc. You need not remember everything. The only thing you'll have to remember is where the SRY gene is present and what is the characteristic of the centromere. Because in the centromere, you are gonna get the questions like with regards to the region of the centromere, whether the centromere is in the center, whether the centromere is in the top, etc. Okay, regarding this, you're going to get some questions. Other than that, the SRY gene is all you'll have to remember in the chromosome chapter. That's it. So now that you know what is the genetic information that is present in a cell, let us discuss with this genetic information, the cell has to form, whether it is going to go into formation of a oocyte or whether it is going to form into a sperm, right? Because the chromosome, the two important chromosomes are the sex chromosome. This sex chromosome decides whether the formation of gonad of a female should occur or the gonad of a male should occur. Let us take the body has or the chromosome has no SRY gene and it has decided to undergo into the formation of a gonad of female, that is ovary. What is the gonad of female? Female gonad is ovary, male gonad is testis. Okay, so now that the body has finally decided, or the cell has finally decided that it has to undergo formation of a female gonad ovary, it has to undergo something known as oogenesis. Okay, so let us discuss about the gametogenesis inside the body. Because to discuss about embryology, the first and foremost thing that you will have to learn about the formation of gametes. That is, in female the oocyte or the egg, and in female that is, and in the male that is sperm. So, before knowing the embryology, which occurs after the fertilization of these two gametes, let us learn about the gametes first, and then we will go into fertilization. Oogenesis. Oogenesis occurs in the female ovary. The progenitor cells tend to go and form the oogonia, then the primary oocyte, then finally forming the secondary oocyte. Okay, so secondary oocyte which undergoes formation of egg and which further it goes into fertilization and forms the zygote. Right. So oogenesis, need not worry about the steps and all because you already would have learnt in your 12th and 11th. So let us discuss more about where the arrests in the oogenesis takes place, etc. Because that is where the problems occur and that is where the new technology of IVF, etc. has been taking place. That is where the competitive exam also tend to move into, because competitive exams have been focused mainly on the clinical significance of certain particular step rather than questioning your remembrance or what your factual knowledge is all about. They are more leaning towards the clinical knowledge or the clinical significance of that particular step. So let us take, for example, in oogenesis itself, they are more interested in knowing whether the doctor knows where should we arrest the particular oocyte so that we can generate particular embryo, etc. Okay, so that is why it is more leaning towards knowing the clinical significance of where the arrest is taking place and rather than knowing the steps of oogenesis or spermatogenesis. So when discussing about the arrest, as I've discussed, the primary oocyte that is formed during the oogenesis will be arrested in the prophase 1 of the diplotin stage. Okay, so the first arrest of the oogenesis takes place in the diplotin stage or the prophase 1. This is pretty confusing, right? So let me make it clear for you. Ovary has something known as primordial germ cells. This undergoes oogenesis to form an egg. In formation of oogenesis, it has to undergo primary oocyte formation, from there the secondary oocyte formation. Okay, so during this formation, the primary oocyte, which is inside the primordial follicle, will undergo one arrest, first arrest. Then the secondary oocyte will also undergo a second arrest. This first arrest will be taking place in the prophase 1. Which particular stage of prophase 1? That is diplotin stage. The second arrest will be taking place in the metaphase 2. Here it is prophase 1 and here it is metaphase 2. You know that, right? In meiosis we have 4 stages, that is prophase, metaphase, anaphase and telophase. This takes place twice. So here in prophase 1, that is first arrest, and metaphase 2, the second arrest takes place. This is very important. These arrests are very important because here the clinical significance of whether the oogenesis is occurring or whether the arrest has occurred. Why is there no oogenesis occurring, or there is no puberty occurring? What PMOS, the new guidelines of PMOS has come up. So which stage is the oocyte or the follicle present in? All this clinical significance is very important. With regards to oogenesis, that is why it is better to know about primary the arrest of the oocytes. Okay, that is all you need to know or remember about here. These are the stages that you need to remember. The other main, now you got to know the stage or the arrest where the oocyte occurs, right? So let us discuss about the polar bodies that are released in the oogenesis. So the first polar body is released 12 hours before the ovulation. The second polar body is released during the 24 hours before the ovulation. Okay, this is first polar body 12 hours before ovulation, and second polar body is 24 hours after the ovulation. This is all you need to remember about oogenesis. At first, we started discussing about the genetic material which decides what gamete the body has to produce through chromosomes, right? Then we went on to discuss about what the gametogenesis is all about. In gametogenesis, now we have discussed about oogenesis and where the arrests occur, because in the new technologies of IVF etc. or PMOS etc., it is important to know where the arrest of the certain particular follicle or the oocyte has taken place. So that is where that is what is important here. So now let us discuss about spermatogenesis. In spermatogenesis, only thing you'll have to remember is, again here also the sperm formation takes place from the primordial germ cells. Okay. So only thing you'll have to remember is about the supportive cells of the spermatogenesis formation, that is Sertoli cell, which gives the nutrition to the spermatocyte or the spermatozoa, the phagocytosis of the excess cytoplasm from the spermatids, and production of inhibin. This is the important function of Sertoli cells, and the important function of Leydig cells is the testosterone formation. Okay, that is all you will have to remember about the pre-embryology or the gametogenesis of the embryology. Further moving on, now that we know that there is formation of oocyte from female gonad and there is formation of sperm from the male gonad. So let us discuss about in detail about embryology. What is embryology? Embryology is the study of embryo. What is embryo? Embryo is the stage of zygote or the stage of fetus from 3rd week till 8th week, okay, of intrauterine life. From 3rd week till 8th week period is known as embryonic period. This is where the gastrulation occurs. Don't worry much about gastrulation, etc. I'll teach you about it. But remember, the third to 8th week is known as embryonic period. And from 9th week onwards till the birth, it is known as fetus. Okay, so that is all you have to remember here. So now we have learned about the gamete formation. Let us discuss about how these gametes undergo fertilization into the formation of an embryo. Fertilization takes place mostly in ampulla. Is it that it doesn't take place in other places? Yes, it will take place, but there is high risk of formation of ectopic pregnancy if it takes place elsewhere other than ampulla. So remember that. So in particular, there is something known as ampulla in the fallopian tube of the uterus. Okay, so that is where the fertilization most of the time takes place. Further, after the fertilization, see, one more thing that I missed here was the arrest that took place in the gonad, that is ovary. The first arrest took place was until puberty. Okay, until puberty, the first arrest takes place. Until fertilization, the second arrest takes place. Once the fertilization occurs, right, the second arrest will undergo the other formation and the follicle will rupture and the secondary oocyte will come out. Okay, so this occurs after the fertilization. The first primary arrest would have taken place before the puberty. Once the puberty is attained, the cyclical changes that occur due to puberty and the hormonal changes that occur due to puberty tend to make the primary oocyte undergo change and formation of secondary oocyte will take place. Okay, remember this. The only thing that you will have to remember here is the first arrest takes place in prophase 1. This will be till puberty. Then the second arrest will take place in the metaphase 2, which will be till fertilization. Once the fertilization occurs, the other changes will start occurring. Okay. So here the fertilization has occurred. Once the fertilization has occurred, the zygote formation starts taking place. That is, from one cell it undergoes two cell. Here, is it undergoing meiosis? No, it is undergoing mitosis. The zygote formation is mitosis. The gamete formation was meiosis. Don't confuse with each other. Okay, so gamete formation was meiosis, whereas zygote formation is mitosis, because here we require 2n to be formed into 2n only. But here we did not. We do not require 2n to be forming 2n. We need 2n to be forming n. That is, what is this n, 2n and all? That is, from 46 chromosome we require 23 chromosome, because the other 23 we are getting it from the other sex, right? One from mother we are getting, the other one we are getting from father. Right, so that is why, and these two combine to form 46 chromosome, which has formed the zygote 46. Okay, so it is undergoing mitosis. It keeps on undergoing mitosis. There comes a stage where the morula formation occurs, and then this morula will be about 16-celled zygote, which undergoes formation of blastocyst. Why blastocyst? Because there is a fluid-filled cavity. Cyst is nothing but a fluid-filled cavity. This fluid-filled cavity inside the zygote stage is known as blastocyst stage, which is in the day 5. You need not remember these days and all. The only important day that you need to remember is the day of implantation. That is day 6 to day 10. That is all you will have to remember. The only important date is day 6 to day 10, the implantation occurs. This is all about the basics of embryology. This is all you will have to remember in embryology basics. Okay, I hope you have understood. First, we discussed about the genetics that is required to formation of gonads. Once the gonads are formed, there is formation of gametocytes. that is oocyte and sperms. So oocyte and sperm combined to form combined or underwent fertilization to form a zygote. Now the zygote has gone on to form blastocyst, which underwent implantation. From now on, we'll start discussing about the further formation of an embryo or a zygote, which eventually forms a baby and which eventually forms a complete human being, which we'll be discussing further. So guys, moving on further, let us discuss now about the formation of embryo. Okay, so now that we know the zygote has implanted, so let us discuss more about the formation of an embryo. So after this implantation, so let me clear you one thing here. So this placenta formation, etc., we'll be discussing more about it during the discussion about obstetrics and gynecology. Now let us focus only about the formation of this particular zygote. Okay, so don't confuse why is he not discussing about the formation of this trophoblast and etc. I'll tell you a little about it, but more about it in the discussion about obstetrics and gynecology, because there itself I'll be discussing about the clinical significance of these things also. So focus only on the particular stuff of the zygote. Okay. So once the zygote has been implanted, there will be a rapid progression of formation of the embryo. Okay. So during this period, there happens something known as gastrulation. What is this gastrulation? Every cell is formed from something known as germ cells. All the organisms, whether it might be dog, cat, everything, all the organism require something known as germ cell, from where the cell differentiation occur and forms into a particular thing. Okay, for formation of hand, let us take formation of eye, everything. This germ cell itself decide what cell has to form into what. So to form the germ cells, the zygote has to undergo something known as gastrulation. So now let us discuss about gastrulation. Okay. People, so my duty is to make your concepts clear and wide, so that you will be able to retrieve your information, whether it is a clinical case that you are dealing with or whether it is a competitive exam that you are dealing with. So that is why let us go in one order that I'll be teaching you. Okay, so please don't confuse everything and make your head go round and round, so that you won't understand anything at all. Okay, so let me be clear with everything that I'm discussing about. First, we discussed about chromosome, right? Chromosome contained so basically chromosome contained the genetic information that the cell required, information that the cell had required. We discussed about the structure of the chromosome. Let's see, close your eyes and start visualizing. If you guys have seen something, one series known as Good Doctor, where the Sean Murphy closes his eyes and starts thinking or visualizing everything in a human body, you will also have to do all this thing while you are studying this 10 days. Okay, so this 10-day series, all you have to do is visualization. So even before knowing about human body, being a doctor, let us start learning about how the human body was formed. So that is why I have taken this chapter first. So now start visualizing chromosome. Okay, chromosome structure we have seen. Chromosome contained a genetic material which that is the gene, the SRY gene, which made the cell to decide whether it has to undergo the formation of a female gonad, that is ovary, or it has to undergo the formation of a male gonad, that is testis. So once that was decided, the oogenesis occurred in ovary, then the testis underwent spermatogenesis. So once this was done, the oogenesis took first and second arrest. After that first and second arrest, this particular oocyte underwent fertilization. Oocyte was formed. Then the spermatogenesis occurred and sperm was formed. This sperm went to something known as capacitation. This capacitation is, what is capacitation? I didn't discuss only, right? So capacitation is to make the sperm able to go and fertilize the ovary. Okay, so this is preparing the sperm to go and fertilize the ovary. This is known as capacitation. More about capacitation when we are discussing about more regarding the male genitals, etc., which will come at the last when we are discussing about the pelvis. Okay, so don't confuse now here itself. At the end of the 10 days, you will be able to visualize and answer anything and everything regarding the human body. Okay, so that is my duty. Okay, so your duty is only to sit and listen, take notes, and come along with me. Okay, don't go in your own way that I'm gonna do this first and then come next with the other topic, etc. Okay, so come along with me. See, that is why I'm giving a flow chart. So okay, the sperm was formed. These two, sperm and the oocyte, decided to undergo fertilization, which underwent in the ampulla of uterus. So then after fertilization, this went on into the uterine cavity, forming morula, blastocyst, etc., and went on to go and implant. When did this implantation take place? 6 to 10 days after the fertilization, the implantation took place. So this much we have discussed, right? Further going down from implantation, we are starting to discuss something known as gastrulation. What is this gastrulation? Gastrulation is nothing but formation of germ cells. See, the zygote formed after the fertilization was nothing but a round body of a cell. It is undergoing rapid mitosis. How does it know whether the body has to form an eye, leg, and a kidney or something like that, right? So for the inside the zygote we have chromosome. The chromosome knows what cell has to form what, etc. But for that cell or for that chromosome to form that particular part of the human being has to undergo something known as gastrulation, which will differentiate the cells to tell them to form what particular organ that has to form. Okay, so that process is known as gastrulation. Okay, nothing but formation of germ cells. This germ cell formation. Okay, so now we'll be discussing about gastrulation first. That is formation of the germ cells, etc. And then after the formation of germ cell, the embryo will start developing into a full-grown fetus. Okay, this embryo will form the systems. Embryo forming the systems and differentiate into one particular fetus. From fetus, after undergoing delivery, this will form an infant. From an infant, it will form an adolescent. In the neonatal formation, everything will happen, and then adolescent to grown old adult. Then aging will occur. Then the death will take place. So this is everything that we are gonna discuss in our chapters. Okay, so you need not worry. I will tell you each and every step in all these 10 days. The fetus formation, the delivery, the infant formation, what are the changes that occurs in an infant, what are the clinical significance in an infant, adolescent clinical significance, what are the cases that we'll see, everything, the adult, the aging, what are the new medicines that have come up for reducing aging, then what happens after the during when there is a death that occurred unnaturally, when there is a natural death occurring, what are the changes that we'll see in our body. We'll discuss everything, the FMT, everything will come around. But please follow the way that I'm teaching you, rather than going into like if I'm discussing embryology, after embryology you going into studying abdomen won't work. Okay, so go in a pathway and I'll clear everything for you. Okay. Okay, so now we were discussing about gastrulation, so let us discuss about gastrulation. So as I told you, so gastrulation is all about formation of germ cell. How does it form? Now that the zygote has formed, zygote being formed, there is one cyst inside the zygote, that is blastocyst, which went on to becoming something known as yolk sac. Okay, you know egg. egg has something known as yolk. What is the function of yolk? Yolk provides nutrition to the particular growing organism, right? So it's the same function here also it provides, okay, yolk sac. So for yolk sac undergoes further differentiation into formation of allantois, etc. Okay, but that we'll discuss further. But now here, just think about it. Now start visualizing whatever I'm gonna tell you. Okay, there is one big ball. Inside one big ball, there is one simple cavity which was the blastocyst cavity, which went on to become a yolk sac. Okay, this yolk sac, okay, this is the ball that you will have to visualize. So this is the yolk sac, and this is the embryo that was being formed. Okay, so there are two types of cells that were formed during this formation. Okay, one is epiblast, the other one is hypoblast. Okay, the lining of the yolk sac was formed by hypoblast. The lining of the embryo was being formed by epiblast. Okay, think it in a three-dimensional structure. Two balls are there. Okay, so two balls. I mean, one single ball was there, but think it as two balls. One compartment that is dividing these two things. One is yolk sac. This yolk sac is being covered by hypoblast. The above thing is epiblast. Okay, so think of this in a 3D or three-dimensional manner. Okay, epiblast. So now after formation of after the implantation, all these changes are taking place to formation of one single embryo. Okay, so once this thing is formed, the epiblast tend to go and form something known as a primitive groove. Okay, this is the primitive groove. You've heard of this so many times in your first year MBBS also. Primitive groove formation will take place. A notochord formation from the primitive groove will take place. All these things, right? What is this primitive groove? Primitive groove is a groove that has happened inside the epiblast. Okay, so let me be clear here. Yeah, so this is a opening, the primitive streak, that is a opening inside this epiblast, okay, which is connecting the epiblast with the hypoblast. So basically here one opening has occurred. Epiblast is there, hypoblast is there, one opening groove is there. So through this groove, what happens is there is migration. The epiblast cell starts to migrate towards the epiblast. So here was yolk sac, right? See, here there was yolk sac, right? This epiblast starts to migrate towards the hypoblast. When this migration starts happening, the above remained epiblast started forming the ectoderm. The cell that was migrated towards the yolk sac started forming the endoderm. Ectoderm was which remained there itself. Okay, so once this has occurred, still further after these two layers were formed, still further the migration of the epiblast take place. Okay, so when still further the migration takes place, there is formation of something known as mesoderm. Okay, once for all, once this gastrulation is formed, the gastrulation is the process. The gastrula is the final product. Okay, once this gastrula is formed, we will get the three germ layers. What are those three germ layers? Ectoderm, endoderm, and mesoderm. These three things you have heard, right? Ectoderm, endoderm, mesoderm, germ cell layers, etc. What are the germ cell diseases, etc. You would have heard that particular term, right? This is what it is all about. Only these three layers. These three layers are the stem cell or the germ cell layers. Okay, ectoderm, mesoderm, endoderm. Everything of the human body is formed by these three cells only. Okay, ectoderm, mesoderm, and endoderm. Only these three things will form everything in our body. So now that you know what the gastrulation was all about, see, let us go to our flow chart again. So here we discussed about gastrulation, where we learned about the formation of germ cell. Germ cell was formation of three layers which undergo differentiation to form an embryo. Right? So now that we have discussed the three layers that will be formed during gastrulation, let us discuss what those three layers will tend to go on differentiation and form into. Okay, this is very important, because in competitive exam definitely you are gonna get this one of this chart questions. Okay, I mean one of the question from these charts. So let us discuss about ectoderm. Ectoderm, as we discussed during the formation itself, it was everything that was outside. See, here also it was outside. The one thing that got migrated went inside. The one that was in between the ectoderm and endoderm was this. This thing, the one in the green, bluish green, is mesoderm. Okay. So now start visualizing about embryo. The one thing that was laid outside, the ectoderm, will go on and differentiate to form the outer epithelium of the body. Everything that is outside the body will be formed by epithelium. Remember that, okay? Epithelium, ectoderm. Whatever is inside, all the epithelium or the inner lining of the body is formed by endoderm. Endodermal epithelial lining in endoderm. For example, let us take GIT. Okay, GIT, gastrointestinal tract, from mouth till anus. Okay, there also you don't confuse the ectoderm, the outer region of the anus. From the anal canal, outer region will be formed by epithelium, because this is the outer structure. The inner region till the dentate line will be formed by the endoderm. Okay, so that is why I am telling you, the inner lining, everything you think of, will be formed by endoderm, because they are inside, endo. Whichever is outside, outer, all the skin layers, epidermal layer, everything will be formed by ectoderm. What is remaining? Middle, mesoderm. All the organs, everything that you know, will be formed by mesoderm. All the renal system, everything, all the smooth muscles, everything, the cardiac system, everything will be formed. Everything that you know regarding smooth muscle system or the muscular skeletal system, everything will be formed by mesoderm. The outer lining of the body is formed by ectoderm. Inner lining is formed by mesoderm. Everything else is formed by inner lining is formed by endoderm. Everything else is formed by mesoderm. That is all you will have to remember. So here is a beautiful chart that I have created with regards to ectoderm, mesoderm and endoderm derivatives. Question is gonna come something like this. Most of the time they'll focus more on mesoderm only, because mesoderm has three different other layers. I'll tell you about, but I'll tell you briefly here. There is something known as paraxial mesoderm, intermediate mesoderm and lateral plate mesoderm. Okay, from these three different parts only all the organs that will the generation will take place. So that is why if there is a defect in formation, there will be an abnormality or anomaly in the formation of that particular organ. Okay, so that is why they tend to more put pressure on mesoderm rather than other. For example, let us take ectoderm. They're not going to discuss more about ectoderm, because it is the outer lining or outer lining of the body. Also, ectoderm is very important with regards to neural crest cells. As I discussed about mesoderm, there are three different parts. Ectoderm also go on to form something known as neural crest cells. Neural crest cells is the main cell that will go on to form the CNS, PNS, etc. Okay, that is why neural crest cell thing is also very important. But questions will be more leaning towards the mesoderm formation. Endoderm, anyway, you know, inner lining of everything is endoderm. For example, if they give the lining of small intestine what is the germ line of the small intestine inner lining? What is it? Endoderm. That's it. But when we are discussing about the what is the germ line of kidney, or what is the germ line of pleura, lung, etc., it will be quite difficult if you are not knowing which particular part of mesoderm it did form from. Okay, so that is why it is important to discuss about this particular chart. So ectoderm, as I told you, everything outside. That is, see, epidermis of the epidermis, hair, nails, this one, sebaceous glands, etc. Everything that is outside, covering, outer ectoderm, epidermis, everything. The lens, cornea, everything is outside, right? Everything will be formed from ectoderm. What else? As I told you, the most important part in ectoderm is neural crest cells, because this is what forms the peripheral nervous system, adrenal medulla, adrenal medulla, melanocytes, facial cartilage, dentine of the teeth. This is very important with regards to ectoderm. Other than that, ectoderm will go and form the CNS from neural tube. I'll discuss about neural tube formation also when we are discussing about CNS embryology. But here, all you need to know is about gastrulation, germ cell formation, basics of embryology, that's it. Okay, so that is why you'll have to know from ectoderm what all was formed, all the outer lining of skin, teeth, eye, in eye, that is the lens, cornea, etc. Then, as I told you, the important differentiating factor here in ectoderm is neural crest cells and neural tube. Neural crest cells form the PNS or the peripheral nervous system, that is ganglion, Schwann cells, etc. Whereas the neural tube go on to form the CNS. Apart from that, apart from CNS and PNS, the electrical system in our body, everything else, all the organs, everything will be formed by mesoderm. Okay, remember this, mesoderm. Then you guys will be questioning, for example, you'll be sitting in a competitive exam, whether it might be INI or NEET. More in INI, these type of questions will be asked. That is, what kind of germ cell was the gastrointestine formed from? Gastrointestine was formed from what? It was mesoderm, right? They're not asking about the inner lining. Inner lining of the gastrointestine was not asked. It was asked what the GIT was formed. GIT was formed from mesoderm. Inner lining was formed from endoderm. Okay, so this was the GIT was formed from mesoderm. Okay, so this is where the confusions will occur, and this is where we will have to focus about. That's it. This is all about gastrulation, what the three germ cell layers will go on and form into. Okay, so now again let us go back into our flowchart. So now that we have learned about gastrulation, formation of germ cell, now it is better to discuss about now the embryo formation has started occurring. That is, gastrulation, the three germ cell formation has occurred till 3 weeks of intrauterine life. See, the fertilization took place, 3 weeks has occurred, all the germ cell layers have formed, and one particular blob of embryo has occurred. Now the real embryo formation will start occurring. Okay, so let us discuss about embryo formation. That is, from 3rd week till 8th week, what are the things that will occur inside one particular embryo? So first and foremost thing that we'll have to discuss during the formation of an embryo is the formation of pharyngeal arches. Okay, this is all about pharyngeal arches. This image is not much of an importance. Okay, don't get confused. What is all this different parts that have been given? See, when you see this embryo itself, now start visualizing in a 3-dimensional way. Think you itself, you are what is his name? Sean Murphy. Okay, think yourself, you are Sean Murphy, and you are visualizing into embryo. So this is the picture that has to come to your mind when you are thinking of an embryo. Okay, so 3rd week has occurred, and now this is the fate of embryo. Now we are going to discuss about pharyngeal arches. Okay, this thing you all would have seen, and everyone has a confusion, including me, until I started learning about embryology. Even I didn't learn anything about embryology in first year. I'm going to be damn truthful, but this is the thing. Even I had many confusion. Only when I started preparing for my competitive exams only, I started learning about embryology. Until then, I also didn't know anything about pharyngeal arches. Also, they are telling some mastication muscle develops from first arch, second arch, third arch, parathyroid gland. I literally didn't know. Only when I started learning in a flowchart manner, I started understanding what embryology is all about. Okay. So you would have seen this image, and we are going to discuss mainly about this particular region of the embryo. What are these? This particular structure is nothing known as pharyngeal arches. This looks like an arch, right? So this is where the pharyngeal part of the body, that is this entire pharyngeal part of the body was developed from this part. Okay, so that is why it is known as pharyngeal arch. Okay, is there only a pharyngeal arch here? There are also cleft side, cleft and arch, cleft and arch. This is an arch, this is a cleft, this is an arch, this is a cleft. So that is why this is known as pharyngeal clefts. Clefts are also there, pharyngeal arches are also there. Okay, so the main thing that we'll have to learn about in pharyngeal arches is all about the number of arches here and what these one single arch will give rise into. Okay, see, this CNS part we'll be discussing in the CNS part we'll be discussing in the head, neck and face development. Head, neck, when we'll be discussing, as I've told you in the previous video, that how we are going to go into, when we are discussing about head, neck and face, once we have completed all the general topics, we'll be discussing about the CNS and TNS formation. When we are discussing about thorax, we'll be discussing about the heart bulge, etc. This is known as heart bulge. Heart bulge, all these things, when the heart starts beating, etc. we'll be learning when we are discussing about thorax. When we'll be discussing about abdomen, we'll be discussing about the formation of gut, the rotation of midgut, what are the anomalies that would occur when there is a failure of midgut formation or midgut rotation. All these things we'll be discussing in abdomen. Then the renal agenesis, etc. we'll be discussing about that in pelvis, etc. Now mainly focus only on what is general to every human being rather than system-wise general. That is what I told in the previous video also. So that is why we are discussing about pharyngeal arches. After the pharyngeal arches, we are done. We are done with general embryology. That's it. According to our flow chart, we are done, right? Now we are undergoing into fetus formation, embryology formation, fetus formation. Here we are. So once we discuss about this, we are done with general embryology. Then we'll start discussing about upper limb, lower limb, etc. Right? So once the general embryology is done, it is done. Don't start thinking about embryology again and again. That's it. Nothing else, no more information about embryology is required. Only this one chart is required. That's it. And this one, this pharyngeal arch chart. In pharyngeal arch, there are 6 pharyngeal arches. Sir, you are telling there are 6 pharyngeal arches, but here there are only 1, 2, 3, 4, 5. Where is the 6th one? Right. So the 5th pharyngeal arch gets dissolved, and only there is 4th and 6th pharyngeal arch. Okay. So no more confusion. 5th pharyngeal arch will be not there. Rather, there will be only 4th and 6th pharyngeal arches. So let us discuss about one by one. So the first pharyngeal arch, remember easy. Each pharyngeal arch, this is a embryo, right? This is in a 2D. Let us turn this embryo, and now there are think of it like this. Okay, here the head is there, and this is the pharyngeal arches. Okay, each of the arch. This is the first arch. This is the second arch. This is third. This is fourth. This is fifth. Both on the right and left side. Both of it is there. This is the image that I am showing here. But once you turn it into the front, this is what it looks like. This is the first pharyngeal arch. Okay, first pharyngeal arch or any arch contains one artery, one vein, and one nerve. Okay, remember that. One artery, one vein, and one nerve. Okay, each of this will go and start supplying one particular thing that these particular arches produce. Okay, let us start discussing about each arch. First arch gives mastication muscles. Okay. Mastication muscles. The first pharyngeal arch started here. Then the second pharyngeal arch started going here. Started developing here. Then stylopharynges here. Then the fourth one, pharyngeal and laryngeal muscles inside. Okay, pharyngeal and laryngeal muscles here. So think of these arches like the parts of face and upper chest. Okay. First pharyngeal arch was mastication. Second pharyngeal arch is all about facial expression. Third pharyngeal arch is all about swallowing, that is pharynx and larynx. Fourth pharyngeal arch, stylopharynx. And then we are discussing about talking. So let us go down. This is pharynx and larynx. And sixth pharyngeal arch is all about the same fourth laryngeal muscles, but except the cricotheroid. Okay, that's it. So these are the 6 parts of the 6 pharyngeal arches. What is the nerve supply? Mastication muscle has that particular nerve supply. Which nerve supplies the mastication? See, when we are discussing about cranial nerves, now only don't confuse. Oh, he's talking about 5th cranial nerve, 6th cranial nerve. No, I'll come back for that also again. But now, for now, remember, once we are done with CNS, right, you will be able to remember the cranial nerves. So once you are able to remember the cranial nerves, and again when you come back and start revising this embryology part of pharyngeal arches, it will be very easy, because once you know the cranial nerves, it is very easy for you to learn about the pharyngeal arches. Because see, 5th cranial nerve, 7th cranial nerve, 9th cranial nerve, 10th cranial nerve, that is all you will have to remember in the pharynx. 5th cranial nerve, what does it supply? Mastication, right? Trigeminal nerve. Trigeminal nerve helps in mastication. So that is done. Second pharyngeal arch, what is it supplied by? Facial nerve. What is the second pharyngeal arch give rise to? Facial muscles. That's it. Then moving on to 9th cranial nerve. What does the 9th cranial nerve do? Glossopharyngeal nerve. Glossopharyngeal nerve, stylopharyngeus, pharyngeal muscles, right? That's it. What does 4th and 6th pharyngeal arch give rise to? Pharyngeal and laryngeal muscles, right? What is it supplied by? 10th cranial nerve. That is vagus nerve, right? That's it. So once you learn about the cranial nerves, it is very easier for you to learn about the pharyngeal arches and what does it give rise to or supply. In the same way, the arteries also. Mastication, maxillary artery. Then we are going down into facial expression. So hyoid and stapedial artery. In the same way. Okay. So very, very, very important topic that is repeated so many times as a question in INI and in NEET also is, and even in your clinical cases also, it will be very useful if you learn this particular fact. That is about the recurrent laryngeal nerve. See, these are the arches. Okay, so these arches, when start developing, these tend to develop the arteries also. So 6th arch on the left side tend to develop the ductus arteriosus. Ductus arteriosus is present. So that is why when the aorta arching happens, the recurrent laryngeal nerve, see, on the right side there is no arch, so that is why there is no recurrence. Okay, recurrence is there. But here it has to undergo in between, below the arch and has to go above. So that is why we will get the recurrent laryngeal nerve. Okay, so 6th arch develops the arch of, 4th arch develops the arch of aorta. So that is why below the arch of aorta, this recurrent laryngeal nerve formation will take place. Okay, so that is how you will have to remember, because in the fourth pharyngeal arch, the arch of aorta was formed, and below that only the laryngeal nerve is being formed. So that is why it has to undergo and come back to serve the laryngeal cartilage, right? Laryngeal muscles, not the laryngeal cartilage, rather than laryngeal muscles. Okay, so this is how you'll have to remember. 4th is above, 6th is below. So when developing the below part, it has to undergo down and come back. So that is how the recurrent laryngeal nerve was being formed. This you'll have to remember. Okay, that's it. Other than that, everything you have learnt about the pharyngeal arches. So let us learn about the important topics that you'll have to learn in laryngeal arches. One is in the formation of pharyngeal arches around 5th week. This is very important. Remember 5th week. Okay, this is the structure you'll be seeing in 5th week, and the competitive exam questions are also mainly based on this particular image. Rather than this image, the questions will be more about this image. Okay, this is around 5th week. Okay, so that is why this is important. So these arches, I told you what they will form into. But what about clefts and sinuses? Okay, so this side, the lateral side is known as cleft. Okay, sorry, sinus. Okay, and this side of the medial part of the side of the arch is known as cleft. Okay, so the first, below the first and second, between the first and second arch, there is first sinus which will tend to go and form the external auditory canal. See, first one was about to form the mastication muscles. Second was about to form the facial muscles. Between that, what is there? Auditory canal is there. So that is what it's all about. Visualization. First one, external auditory canal. You simply go on going on and mugging up things won't work. Okay, here. So start visualizing. So here the mastication muscle was there. Here the facial expression was there. In between that, external auditory canal was formed. Then again here, third pharyngeal arch and fourth pharyngeal arch. The pharyngeal muscles and laryngeal part was there. Stylopharyngeus and laryngeal part was there. Between that, here one sinus is there. If the sinus is not closed, something known as cervical sinus will be formed. If the cervical sinus persists, then that will go on and tend to form brachial cyst. Okay, this is very, very important. This is one of the congenital malformation that you will see in a fetus, which is known as brachial cyst. This is because if the cervical sinus persists, that is between third and fourth, that is done. Sinus part of the pharyngeal artery is done. Then moving on to this side, the cleft part. The between first and second, auditory canal was on the outside. Inside was the same canal, right? Eustachian tube. That's it. Auditory canal was there. Continuation of that, eustachian tube. Auditory canal between first and second arch. Eustachian tube on the first and second arch. Then below eustachian tube, what is there? Palatine tonsil, right? Below eustachian tube, inside what is there? Palatine tonsil. That's it. Remember, on the other side, in the second pharyngeal arch, there is palatine tonsil. Then going on below. After the formation of palatine tonsil, between third and fourth, what is that? Thyroid and parathyroid, right? See, remember, thyroid, from the formation will takes place at the level of tongue and then it goes below, right? So that is why the thyroid will be below, okay, after the formation. But the formation of parathyroid is what is important here between third and fourth pharyngeal arch rather than thyroid, because thyroid will be forming near the tongue, right? So parathyroid gland will be here. This particular question is very, very important when you are solving MCQs. This has been asked multiple times in all the MCQs. So where does the parathyroid gland formation takes place? It is between third and fourth pharyngeal cleft. That's it. Then the thymus formation. Thymus, which undergoes degradation, which is a like a lymph node like structure, will be degraded when we become adult. So the thymus formation will take place in the same third and fourth pharyngeal, between the third and fourth pharyngeal cleft. Okay. These three are very, very important. That's it. In this image, no. First you got to know auditory canal and eustachian tube. Below that, what was there? Tonsil. Below the tonsil, thyroid will formation will take place near the tongue, but it will go and lie near the thyroid cartilage. But even before thyroid was being formed, what was there in third and fourth? Parathyroid gland. With parathyroid gland, what is there? Thymus is there. That's it. That is all you'll have to remember about this image. This particular image is all about what I discussed above with regards to formation of cranial nerves and the particular muscles that the cranial nerves supply. That's it. This is everything you need to learn about general embryology. That's it. So, with regards to flow chart, we have completed genetics, the chromosome part, the formation of gonads, formation of gametocytes, then capacitation was discussed, then the fertilization occurred in ampulla, then implantation occurred from 6th to 10th day, then after implantation gastrulation, that is formation of germ cell took place. Germ cell layers are 3 layers, that is ectoderm, mesoderm and endoderm. From these three, ectoderm, endoderm and mesoderm, embryo formation or the differentiation took place into one particular organ or particular lining. Then after the formation, we discussed about fetus formation or the embryo formation itself after the gastrulation. During fetus formation, we started discussing about pharyngeal arches. Rest all the structures, either it might be CNS, heart development, lung development, each of these systems will be discussed when we are discussing about the formation of, when we are discussing that particular system. That's it.